Hi there, science fans. Welcome to another Science and Other Stuff for Senior Citizen video. In the present video, I will introduce you to the medical and clinical discipline of immunotherapy. Research and clinical application of immunotherapy has advanced during the last decade to the point of being a major contributor to the path to curing cancer. So what is cancer? Cancer is a disease in which some of the body's cells grow uncontrollably and may spread to other parts of the body. This latter event is termed metastasis. In the graphic, panel A presents an illustration of a section of an organ where the cells are contained and their morphology or the individual shapes are defined and orderly. The different normal cells are also contained within a basement membrane, separating one tissue from another. In panel B, the abnormal cancer cells have a very different shape and are increasing their number by uncontrolled cell division. Additionally, the basement membrane no longer serves as a barrier, and the abnormal cancer cells are invading the surrounding tissue, beginning the process of metastasis to other organs. This process is facilitated by passage into blood vessels. If you are interested in a video describing how normal cells are transformed to cancer cells and the mechanism by which that process occurs, please leave me a comment in the comments section. In the past, there were primarily three conventional methods for the treatment of all cancers. In the case of solid tumors that are encapsulated and have not metastasized to other regions of the body, surgical removal of the tumor is generally the first treatment of choice. In many instances, the tumor location may not be amenable to surgery alone, but requires the use of a combined treatment such as radiation therapy and/or chemotherapy. There are several disadvantages to Method 1, including the possibility that the location of the tumor is too close to vital blood vessels, the possibility of not excising the entire tumor and leaving some cancer cells behind that can produce a tumor, the possibility that some cancer cells may have escaped the confines of the tumor and seeded in other organs. And surgery is an invasive method with complications associated with any surgical procedure. Many cancers are cancers of the blood, so surgical procedures are not applicable. Radiotherapy may be used in the early stages of cancer or after it has started to spread. It can be used to try and cure the cancer completely, or can be combined with chemotherapy or used prior to surgery. Radiotherapy involves giving high doses of radiation beams directly into the tumor to break the DNA of cancer cells within the tumors in order to decrease further growth of the tumor and causing it to shrink or die. In the case of prostate cancer, radiotherapy is 95% effective. Radiotherapy may not kill all of the cancer cells, and those remaining can regrow into a tumor. Side effects include tiredness and diarrhea, and normal cells close to the tumor may become damaged and die. Since cancer is a disease in which some of the body's cells grow uncontrollably and may spread to other parts of the body, certain powerful drugs that inhibit cell division are used to kill cancer cells. Chemotherapy is a type of cancer treatment that uses drugs that kill fast-growing cancer cells. These drugs circulate throughout the bloodstream, thereby treating cancer cells almost anywhere in the body. Unfortunately, these drugs are not specific to targeting just cancer cells, but also target fast-growing normal cells, such as normal blood cells and normal cells that line the intestine. Some results of this lack of specificity include hair loss, infections, anemia, loss of appetite, and diarrhea. During the last decade, a fourth method, immunotherapy, has been undergoing development and is maturing to the point that some of its products have been approved by the FDA and others are undergoing human clinical trials. So what is immunotherapy? It is an application of the fundamental research of cancer immunology and is a growing subspecialty of oncology. Cancer immunotherapy is the stimulation of a patient's own immune system to treat cancer. These specific immunotherapeutics are designed to train and improve the immune system's natural ability to fight disease. Before getting into an in-depth discussion of the types of immunotherapies, we need to review some basic immunology. Defenses against viruses, microbes, foreign substances, as well as invasion by a patient's own cancer cells is facilitated by sequential and coordinated responses called innate and adaptive immunity. Innate immunity is essential for defending against foreign bodies in the first few hours or days after an infection, before adaptive immune response have developed. Cells of the innate immune system are in place even before an infection occurs. Macrophages engulf microbes or foreign bodies and destroy them within the cellular boundaries of those macrophages. Natural killer cells recognize infected cells and cancer cells and kills them. Cells of the innate immune system also produce chemicals and cytokine protein signals used to recruit other immune cells to help and eradicate the invading enemy. Adaptive immune responses develop later and require the activation of lymphocytes. Examples of innate and adaptive immunity are presented here. Here we see a graphical representation of parts of the innate and adaptive immune system responding to an invasion of the body by foreign pathogens, as depicted in Panel A, Step 1. The foreign invader is presented here as antigens. Antigens are proteins located on the surface of the foreign invader as well as inside the cells of the invader. These antigens are detected by macrophages, NK killer cells, dendritic cell precursors, as well as other innate immune cells. These cells proceed to destroy the foreign bodies and also secrete cytokines that act as signals to recruit more of these innate immune cells. In Step 2, the precursor dendritic cells develop into immature dendritic cells that capture the antigens and display them on their cell surface. As these immature dendritic cells mature, they migrate from the blood to lymph nodes, where they present the antigen to B and T lymphocytes. This is represented in Steps 3, 4, and 5. In Panel B, the process continues through the activation of T and B cells that continue to mature through differentiation and proliferation to the formation of B memory cells and plasma cells that make antibodies that are specific to the particular antigen associated with the foreign invader. The T cell branch of the activation, maturation, and proliferation wing of this process results in the formation of a variety of cells, including T killer cells and T memory cells. The combination of the innate and adaptive immune system protects us from invading pathogens, as well as forming B and T memory cells that remember that particular antigen and will be activated again if we are invaded by that antigen in the future. This combined system is the same process that is used to protect us from abnormal cancer cells that may occur in our body. So how does the immune system discriminate between our own normal cells and our own abnormal cells? It does this by detecting abnormal antigens that are expressed on the surface of our cancer cells, as well as antigens that are located inside of our cancer cells. Our cells are defined as normal during our residence time in utero. We had immune surveillance cells, as depicted in Panel A of this graphic, survey our cells and stamp them as normal. The antigenic signature of our normal cells are remembered by our immune system, and if our immune system is behaving properly, our innate and adaptive immune system does not attack and destroy our normal cells. However, if our normal cells become abnormal cancer cells, our immune system recognizes these cells because of specific non-self antigens, and in the case of cancer cells, these non-self antigens are tumor antigens, as shown in Panel B of this graphic. As already mentioned, immunotherapy is a treatment that uses the person's own immune system to fight cancer. Immunotherapy can change or boost how the immune system works so it can find and attack cancer cells. At times, a person's immune system underperforms. This may be due to a variety of factors, including simply being overwhelmed because the tumor is so very aggressive, or because of the immune suppression, or because the cancer cell has evolved to fool the immune system into thinking that the cancer cell is a normal cell. Several types of immunotherapies have been developed and are presently being used in cancer patients. These include infusion of patients with monoclonal antibodies, use of checkpoint inhibitors, vaccines, T-cell transfers, and the use of cytokines. These immunotherapies will be presented in the following slides. This graphic illustrates the types of antibodies that are either already in use as immunotherapeutics or are in different stages of development. Monoclonal antibodies are proteins made in a laboratory and are used to stimulate the immune system to destroy pathogens or cancer cells. Monoclonal antibodies are used to interfere with specific receptors on a specific cancer type, such as the HER2 receptor on certain breast cancers. They are also used to interfere with receptors on activated T cells and receptors on cancer cells called checkpoint. We will present examples in the following graphics. Vaccines are also in the pipeline of tools to inactivate and kill cancer cells. Vaccines can be made from cancer cell proteins or fragments of proteins called peptides. Vaccines can also be made using the technologies developed for decreasing infectivity and pathology of the COVID-19 virus. These technologies include the use of inactivated DNA viruses, like those used by Johnson & Johnson's COVID-19 vaccine, and the mRNA technologies used to make the Pfizer and Moderna COVID-19 vaccines. This graphic indicates how monoclonal antibodies are presently being used to treat certain breast cancers. HER2 is a cancer gene that makes the protein human epidermal growth factor receptor 2. The gene is involved in normal cell growth. However, the gene can be overexpressed in some cancers. HER2 is overexpressed in 15 to 30% of invasive breast cancers. Breast cancers can have up to 25 to 50 copies of the HER2 gene and up to 40 to 100-fold increase in HER2 protein, resulting in over 2 million receptors expressed at each tumor cell surface. Herceptin-2 is a monoclonal antibody specifically designed to target HER2 receptors. It works by blocking the effects of HER2 and encouraging the immune system to attack and kill cancer cells. Panel A represents a graphic indicating the number of surface HER2 receptors on normal breast cells and on HER2 positive cells. The low numbers of HER2 receptors on normal cells result in normal growth and cell division, while the abnormally high numbers of the HER2 receptors in the cancer cells results in uncontrolled growth and cell division. The upper part of panel B shows the mechanism of uncontrolled growth. The HER2 protein signals other like cells to divide and grow into a tumor, which then produces vascular endothelial growth factor that signals growth of blood vessels to infiltrate the tumor and provide additional nutrients to maintain and facilitate increased growth of the tumor. Herceptin, a monoclonal antibody grown in the laboratory, binds to the HER2 receptors, blocking HER2 signaling, thereby preventing cancer cells from multiplying and forming a tumor. Researchers have also developed a monoclonal antibody to vascular endothelial growth factor in order to prevent blood vessel growth. This latter monoclonal antibody is used in some eye diseases. This graphic illustrates how a cancer cell can escape the killing action of the immune system. This escape process occurs as the cancer cell adjusts to the cellular immunity process and learns to suppress the immune system. Panel A portrays how an activated T cell recognizes a cancer cell. T cell activation is described in earlier graphics discussing the innate and adaptive immune system. One can pause this video and review that section if needed. The activated T cells displays a specific T cell receptor, TCR, for the specific cancer. Upon recognition, the TCR protein binds to the MHC protein of the cancer cell and proceeds to kill the cancer cell. However, over time, the cancer cells learn how to suppress this recognition killing process. Panel B describes the suppressive process. The cancer cells express a protein on its surface called PD-L1. The cancer cell PD-L1 protein binds to the activated T cell surface protein PD-1 receptor protein located on the activated T cell and inactivates the T cell killing function. This inactivation occurs because the T cell was fooled into designating the cancer cell as normal self. Some immunotherapy drugs are monoclonal antibodies called checkpoint inhibitors that work by blocking proteins like PD-1 from binding with their partner proteins PD-L1. This is depicted in panel A of the graphic. This action prevents the off signal from being sent to the activated T cell, allowing the T cell to kill the cancer cell. This is presented in panel B of this graphic. T cell transfer therapy is a type of immunotherapy that makes our own immune cells more able to attack cancer. There are two primary types of T cell transfer therapy: tumor-infiltrating lymphocytes, or TIL therapy, and CAR T cell therapy. Both involve collecting a patient's own lymphocytes, growing large numbers of them in the laboratory, and then giving them back to the patients through infusion. Panel A in this graphic shows an image of a tumor containing macrophages and lymphocytes that have infiltrated the tumor in order to destroy the cancer cells. In panel B, the tumor is excised, and the lymphocytes are separated from the tumor tissue and tested in order to determine which ones best recognize the tumor cells. Then these selected lymphocytes are inoculated into Petri plates with tissue culture medium supplemented with growth factors like IL-2 stimulating the growth and proliferation of the activated T cells. The expanded lymphocytes are then infused back into the patient. The reason for this approach is that the lymphocytes in or near the tumor have shown the ability to recognize the tumor or to overcome the signals that the tumor is releasing to suppress the immune system. Given the patient's large numbers of the lymphocytes that reacts best with the tumor may help to overcome these barriers and slow the growth or inhibit tumor growth. CAR T cell therapy is a type of treatment in which a patient's T cells are changed in the laboratory so they will attack cancer cells. T cells are removed from the patient's blood, and then a gene for a special receptor that binds to a certain protein on the patient's cancer cell is added to the T cells. This special receptor is called a chimeric antigen receptor. Large numbers of the CAR T cells then are grown in the laboratory and given back to the patient via infusion. Then the engineered CAR T cells can bind to cancer cells and kill them. CAR T cell therapy is being successfully used to treat certain blood cancers, and human clinical trials are ongoing for treatment of solid tumors and metastatic tumors. Lastly, cytokines are man-made versions of some proteins that boost the immune system. Personalized medicine is now possible because of the recent developments in genomic, proteomic, and metabolomic technologies. Research over the last 10 years has provided medicine with a clearer understanding of how the immune system works. This understanding allows physicians the ability to prescribe drugs that can boost the individual's immune system to fight and conquer foreign invaders such as viruses and bacteria, as well as one's own cancer cells. This graphic represents where the field of immunotherapy is at present and points to future directions in the treatment of cancer using immunotherapy. We can readily isolate DNA and RNA from any normal cell or cancer cell. Sequencing determines the order of the letters A, T, G, and C in the DNA and dictates the kind and function of the specific protein produced. Once that sequence is obtained, use of bioinformatic algorithms can direct us to cancer antigen sequences that distinguish self-antigens from non-self-antigens. This is shown in steps 1 and 2 in this diagram. These non-self or cancer antigens can then be presented to dendritic cells and other antigen-presenting immune cells. Presentations of these antigens to naive T cells activates them, allowing them to be amplified to millions of activated T cells capable of killing a specific cancer cell. This is represented in steps 3, 4, and 5 of this graphic. In step 6, these activated T cells are given back to the patient as an injection or as an infusion. If you like this video, please like, share, subscribe, and ring the bell to be informed of future videos. If you like a clarification of any topic discussed in this video, please leave a comment. If there are areas of medicine or science you'd like a video presented on, please leave a comment. Bye.